I'm Daria Hirschmann, and I'm a PhD student at the Limes Institute in Bonn, where I research hepatic macrophages. I'm co-first author of the publication Kupffer cells control neonatal hepatic metabolism via IGF-1 signaling. And in the next 2 minutes, I'm going to explain to you what we found and why it matters. In our study, we show that hepatic macrophages, also known as Kupffer cells, are particularly important for liver metabolism at birth because they produce insulin-like growth factor 1, IGF-1, which regulates glycogen storage. For me, the most fascinating aspect is that a cell that is classically viewed as an immune cell does not only function as one, but also has a role in tissue development. And this also highlights that we as researchers need to stay open-minded and not define a cell solely by its best known function, but also look out for other more diverse functions. Our findings add a more detailed understanding of how neonatal organs adapt, develop, and what part macrophages play in these processes. We already know that during adult life, macrophages shape their local tissue environment, but much less is known about early development. So here we come in and we show that the Kupffer cells regulate glycogen storage in the neighboring hepatocytes, highlighting that this interaction axis is already very important during early development. My favorite figure is figure 6, particularly the electron microscopy images, and also the quantifications that we draw from these images. There's something very compelling about just seeing cellular changes on an actual image and not deriving them from some sort of obscure machine that you don't understand. So here, also a big shout out to our microscopy core facility because they did an amazing job in helping me acquire those images, which also later on made the journal's cover. Looking into the future, we hope that our findings encourage other researchers to think of macrophages as active regulators of tissue development and metabolism, and not just as mere phagocytes or rubbish collectors. We also hope to continue our research in how the macrophage-tissue interaction shapes the overall organ development and metabolism.